morning, students. I am Pradeep Hirapure, and today I am going to deliver my lecture on the topic history of microbiology. So, as you know that in your microbiology related paper, there is a first unit, and in that unit, there is a first point, that is a development of microbiology. So, we try to discuss here the history of microbiology and what are the different types of researchers, scientists who contributed to in the development of microbiology. So before going to discuss about the contribution of various scientists in the development of microbiology, you should know about the definition of microbiology. Dear students, you know very well this microbiology nothing but the study of microorganism. So the branch of science which dealing with the study of microorganism known as a microbiology. But again question arise in our mind, what is actually the microbes or microorganism? So you know very well, dear students, in the universe there is a different types of living organism, and each and every living organism made up of the living cell. It is a cellular organism, right? But when we talking about the microbiology, nothing but the study of microorganism, it means that the organism are such type of organism which are unable to visualize by our naked eye. Okay, so the organism which are not visualized by our naked eye, and for the visualization, for the observation of that organisms, we need the magnifying glass or microscope. Such type of microorganisms or such type of organisms considered as a microorganism. Now you know very well, in a 12th science you studied about the different forms of the microorganism. It may be algae, it may be fungi, it may be the bacteria, it may be the protozoa. So many different types of microorganisms found in everywhere. So before the discovery of microorganism, the people die. At that time, the people thinking about that, the death of human beings just because of the God punishment. But after the discovery of microorganism, the people considered and understand the every diseases caused by the pathogenic microorganism. It may be the protozoa, it may be the virus, it may be the bacteria or fungi. So who first time observed this microorganism? So as you know that the first scientist, that is Anton Leon Hawk, observed the microorganism under the magnifying glass. But at that time, the scientist Anton Leon Hawk do not use the microorganism word. He used the animalcule. So that animalcule word used for the microorganism by the scientist Anton Leon Hawk. And from that day, we are considered in our environment there are some microscopic organism, and they are responsible to cause the various types of disease. Not only the microorganism always cause the disease, but also the some useful microorganisms also present in our environment, and they are helping to human being to develop the different types of product. It may be the medicinal product, for example the Penicillium notatum. This one is the microscopic organism, that is a fungi, and from that fungi we developed the penicillin, that is the antibiotic. So such type of various types of molecules we can derive from the microorganism and utilize for the human welfare. Okay, so when we talking about the history of microbiology, the one renowned personality in the microbiology world, that is Louis Pasteur. And as per your syllabus, the first scientist is there, that is a Louis Pasteur. So I would like to explain what are the contribution of Louis Pasteur in the development of microbiology. Dear students, this Louis Pasteur born on December 27 in 1822, and he is a France scientist and a French chemist as well as the microbiologist, who was one of the most important founder of medical microbiology. Pasteur contribute in the science and technology as well as the medicine are nearly without precedent. He pioneered the study of molecular asymmetry, discovered that microorganisms cause fermentation and disease originated by the presence of microorganism. Louis Pasteur, well known, recognized by the process that is pasteurization. So now we are going to discuss about the contribution of Louis Pasteur in the development of microbiology. Dear students, you know very well this Louis Pasteur, nothing but the father of modern biotechnology as well as father of microbiology. So the Louis Pasteur play an important role to explain the molecule which showing two different types of symmetry. The same molecular structure of any molecule, but even though that similar structural molecule shows the different symmetry, and this science known as a molecular symmetry. So soon after graduation from the university, the German chemist discovered or explained the molecular symmetry, and who had shown that the tartarate and paratartarate behave differently toward the polarized light. So tartarate rotated the plane of polarized light, whereas paratartarate did not rotate in presence of the polarized light. This was unusual because the compound displayed identical chemical properties, and the Pasteur noted that the tartarate crystal exhibited the asymmetric form of that corresponded to their optical asymmetry. He made the surprising observation that the crystalline paratartarate consisted of a mixture of crystal in a right-handed configuration. However, when these crystals were separated manually, he found that they exhibited right and left symmetry. In other words, a balanced mixture of both right and left crystal was optically inactive. Thus, The Pasteur discovered the existence of molecular asymmetry, the foundation of stereochemistry, as it was revealed by optical activity. Over the course of that next 10 years, Pasteur further investigated the ability of organic substances to rotate the plane of polarized light. He also studied the relationship that exhibited between crystal structure and molecular configuration. His study convinced him that asymmetry was one of the fundamental characteristics of living matter. So dear students, the Pasteur first time discovered that the molecular asymmetry. Now what is molecular asymmetry? You know very well, in the chemistry we studied the some molecule having ability to rotate in two different types of direction. When we pass the polarized light through that molecule, the molecule can rotate in right-handed or left-handed. So these molecules structurally similar. You can see on the slide both the molecule. Here also you can see the COOH group is there, then there is alkyl group is there is a NH2 group, and this central carbon atom and hydrogen atom. So all these atoms also present in this molecule also, but these two molecules shows the different rotation in presence of the polarized light, and this one is the molecular asymmetry. So this phenomena, this type of observation first discovered by the Louis Pasteur. Louis Pasteur again contributed in the fermentation process. This Louis Pasteur first discovered the germ theory of fermentation. In 1854, Pasteur was appointed as a professor of chemistry and dean of science faculty at the University of Lille. While working at Lille, he was asked to help solve problems related to alcohol production at a local laboratory, and thus he began a series of studies on alcoholic fermentation, and his work on these problems led to his involvement in the tackling a variety of other practical and economic problems involving in the fermentation. His efforts proved successfully in unraveling most of these problems, and new theoretical implications emerged from his work. Pasteur investigated a broad range of aspects of fermentation, including the production of compounds such as lactic acid that are responsible for the source of milk. He also studied the butyric acid fermentation. In 1857, Pasteur left Lille and returned to the Paris, having been appointed manager and director of scientific study at the École Normale Supérieure. That same year, he presented experimental evidences for the participation of living organisms in all fermentative processes, and shows that a specific organism was associated with each particular fermentation, and this evidence gave rise to the germ theory of fermentation. After this, the Louis Pasteur revolutionized the dairy industries by inventing the process of pasteurization. This process involves the killing of certain pathogens or microorganisms which are present in the milk in order to preserve the milk. The process of pasteurization kills some but not all microorganisms, only the ones that could be harmful upon the consumption. So when we heat the milk at 55 to 60 degrees Celsius temperature, at that temperature most of the microorganism killed, and this process known as a pasteurization. This pasteurization process discovered by the scientist Louis Pasteur. The Pasteur successfully remove or kill the microorganism from the wine. At that time, with the help of fermentation process, the Pasteur able to produce the alcohol, but that alcohol shows a bitter taste just because of due to the presence of some microorganism in that wine or in that beer. So after the discovery of pasteurization, the Pasteur gives the solution to remove or to kill the microorganism from the wine or beer. And this methodology used regularly in brewing industry. He also devised a method for the manufacturing of beer that are prevented deterioration of the product during the long period of transport of ship. Dear students, this Pasteur again involved in the spontaneous generation. At that time, there so many different types of scientists involved in the controversy of spontaneous generation. So some scientists believe that the life form generated from the non-living things. But the Louis Pasteur in 1859 performed one experiment and tried to explain with this experiment the every living form present on the earth or in the environment originated from the living things, not from the non-living things. So in the fermentation and putrefaction were often perceived as being spontaneous phenomena, and the Pasteur performed the experiment in the laboratory to disprove the spontaneous generation theory, in which he prepared the nutrient medium, liquid nutrient medium, and pour into the one flask. And this nutrient medium heated or boiled with the help of this burner. After that, in one flask, he closed the mouth of that flask with the help of cotton or cotton plug, and the another flask which having a guzan type of neck, okay, and all these three and one flask remain opened here, okay. So in every in all the these three flasks there is a nutrient medium, and all the flasks heated by the flame and boil the nutrient medium, okay. Why? because due to the boiling, whatever the microorganism is there, they are killed. Okay, so this one is the sterilization process. So this nutrient medium now free from the microorganism when we heat to that nutrient medium. After that, these 3 flasks keep into the incubator at 37 degrees Celsius temperature, and after the incubation period he observed that the contamination of bacteria in open flask, but there is no any contamination observed in remaining 2 flasks. Why? Because the flask already closed with the cotton plug. Okay, and that's why the air actually come inside the flask, but whatever the microorganism present in that air, all the microorganisms are filtered out with the help of this cotton plug. And from that day we use the cotton plug as a filter in the microbiological experiment. And in this also there is no any contamination observed, because the flasks having a guzen neck. Okay, so what happened, or what are the mechanism? Due to the presence of this guzen neck, whatever the air actually pass through this guzen neck into the bottom side of flask. So this one is the nutrient medium, and the air actually passes through this guzen neck. At that time, inside there, whatever the dust particle is there, so actually microorganism present in air, we can say inside the air there is a dust particle, and on the surface of dust particle actually microorganism present. So whenever the environmental air passes through this flask toward the bottom side of that flask, all the dust particles actually are there at the surface of this neck. Okay, and only without dust particle the air actually come inside the flask toward the bottom side. So in this flask also the air is coming, but this air free from microorganism. Okay, and that's why in this nutrient medium there is no any bacterial contamination, microbial contamination. And this experiment disprove the spontaneous theory, because here is microbe actually start to grow just because of the environmental microorganism come inside, and due to the presence of nutrient medium that microorganisms start to grow, and we observe the bacterial or microbial contamination in this flask. But here, due to the absence of external microorganism, there is no any microbial growth. It indicates that every living cell originated from the living things, not from the non-living things. Okay, so this one is the disproof of spontaneous theory of spontaneous germ theory. You can see here due to the heating there is no growth. Now this one is the neck, but when we cut this flask through from here, so this nutrient medium exposed in the contaminated air, and that microorganism come inside the nutrient medium and grow, and that's why the the color of nutrient medium changed. Dear students, the Pasteur also work for vaccine development. Now you know very well the vaccine nothing but the attenuated form of pathogenic microorganism. It may be the virus or it may be the bacteria. In the early 1870s, Pasteur had already acquired considerable renown and respect in the France. And in 1873, he was elected as an associated member of Académie de Médecine. The medical establishment was reluctant to accept his germ theory of disease, primarily because it originated from a chemist, because the Pasteur is basically chemist. So however, during the next decade, the Pasteur developed the overall principle of vaccination and contributed to the foundation of immunology. Pasteur's first important discovery in the study of vaccination came in 1879 and concerned a disease called chicken cholera. The Pasteur said that the chance only favor the prepared mind, and it was chance observation through which he discovered that the culture of chicken cholera lost their pathogenicity and retained attenuated the pathogenic characteristics over the course of many generations. And he inoculated the chickens with the attenuated form and demonstrated that the chickens were resistant to the fully viral strain. From them, the Pasteur directed all his experimental work toward the problem of immunization and applied this principle to many other diseases. Pasteur began investigating the anthrax in 1879. At that time, an anthrax epidemic in France and in some other part of the Europe had killed a large number of sheep, and the disease was attacking humans as well. The German physician Robert Koch announced the isolation of anthrax bacillus, which Pasteur confirmed. Koch and Pasteur independently proved the definitive experimental evidences that the anthrax bacillus was indeed responsible for the infection. This firmly established the germ theory of disease, which then emerged as the fundamental concept underlying medical microbiology. Pasteur also contributed to discover the mysterious disease in silkworm. In 1862, the Pasteur was elected to the Academy of Science, and the following years he was appointed as a professor of geology, physics and chemistry. Shortly after this, Pasteur turned his attention to the France silkworm crisis. In the middle of 19th century, the mysterious disease had attacked French silkworm. The silkworm eggs could no longer be produced in France and they could not be imported from other countries. Since the disease had spread all over the Europe and had invented the consequence of that disease. By 1865, the silkworm industry was almost completely ruined in France and to a lesser extent in the rest of Western Europe. The Pasteur knew virtually nothing about the silkworm. But upon the request of his former mentor, Pasteur took charge of the problem and accepting the challenges and seizing the opportunity to learn more about the infectious diseases. He soon became an expert silkworm breeder and identified the organism that caused the silkworm disease. After 5 years of research, he succeeded in the saving of silk industry through a method of enable the preservation of healthy silkworm. The healthy silkworm eggs and prevented their contamination by the disease causing organism within a couple of years. This method was recognized throughout Europe and it is still used today in silk producing countries. So in the next lecture we will discuss about the contribution of Robert Koch